Persistent chemicals are perhaps transiently or maybe even permanently altering the microbiome depending on the exposure window. So one of the big questions people have is if you're exposed early on in life, at birth, does this perhaps set you up in the wrong trajectory, if you will, health-wise?
Welcome to Growing Impact, a podcast by the Institute of Energy and the Environment at Penn State. Each month, Growing Impact explores the projects of Penn State researchers who are solving some of the world's most challenging energy and environmental issues. Each project has been funded by the Institute's Seed Grant program that grows new research ideas into impactful energy and environmental solutions. I'm your host, Kevin Slyman. Perfluorinated substances, also known as PFAS, are man-made chemicals found in a wide variety of everyday items. They've been around since the 1940s, and widespread detection and public awareness increased significantly around the turn of the century. There is growing evidence of links between PFAS exposure and various health effects. And these forever chemicals can now be found in many living creatures, including humans. On this episode of Growing Impact, we discuss a past seed grant on environmental contaminants through which researchers have continued to develop new areas of investigation. One of the important questions raised by this team of researchers is, do PFAS impact the human microbiome? Which are the microorganisms that live in a human's gut? And if so, what's the impact? Hello, Andrew. Hello, Costas.
Hey, Kevin, how are you?
Hi, how are you?
I'm doing great. Thanks so much for being on Growing Impact and discussing your research. Could each of you take a moment and introduce yourselves? Can you tell us your name, your title, and provide a brief background on your research?
I'm Andrew Patterson. I'm the John T. and Paige S. Smith Professor in the College of Agricultural Sciences. My home department is the Department of Veterinary and Biomedical Sciences. I've been here at Penn State now since 2011, and my research really focuses on the chemical communication between the hosts, in most cases mouse models, but also humans, and their microbial partners, so the microbiome. So we've been really active in this area trying to dissect how we communicate with those microbes and vice versa. And so this has been the central focus of our research for, the last 13 years or so. And hence kind of the strong connection that I've had with Costas to really focus on kind of this important question. And that is around how microbes are utilizing substrates that the host provides and vice versa.
So my name is Costas Moranis. I'm the Donald B. Broughton Professor of Chemical Engineering. My home department is Chemical Engineering Department. And our research work straddles the world of biotechnology, biophysics, and biochemistry with the world of computations, optimization, and algorithms. So what we are trying to do is figuring out ways of making metabolic pathways more in tune with your biotechnology objective, finding ways to engineer enzymes to do appropriate bioconversions, engineer antibodies so they recognize specific antigens, etc. So everything that we do in our group is computational. So we are partnering with other labs either at Penn State or elsewhere to help us support our investigations.
Your work on environmental contaminants and their impact on human health has continued to grow. Can you tell us about the start of this effort and what inspired you to research the subject?
When I first came here to Penn State, one of my big interests has always been on understanding how chemicals that we're exposed to impacts us. And we had really only ever focused on this from the host perspective. So really thinking about this, how do these chemicals impact us directly? But during my postdoc, when I was with Frank Gonzalez at the National Cancer Institute, we had some hints that it was impacting other things like the microbes that resided in us. But we really didn't have a lot of the tools and technology and, more importantly, the expertise to really investigate this in a lot of detail. So that was one of the very first things I did when I got here to Penn State was to do some basic sequencing experiments. This is now, probably right after I got here, so 12, 13 years ago. And that was to look at the community changes that were occurring after this exposure. In this case, it was a different drug. It was not an environmental contaminant. But we very quickly saw that there was a big impact on the composition of the microbes, in this case, in the guts of mice. And that very quickly led us to try to focus on what that meant functionally. And so we started combining what I would say is our lab's expertise, and that's metabolomics. And so this is just a way of comprehensively profiling all the small molecules that are in a sample. So glucose, TCA cyclointermediates, bile acids, that sort of thing. And from there, we began to expand upon, any other different type of chemical that we may be exposed to. And so because we're in the Institute of the Environment and Energy, one of the things that we've been focusing on was, thinking about these environmental chemicals. PFAS, perfluorinated species, were on on everyone's radar, even now more than 10 years ago. And now currently it's, hugely important. But beyond that, also looking at other environmental chemicals, persistent ones, dioxins, furans, PCBs, these types of chemicals that we know to be exposed to on a daily basis. And one of the big things we began to wonder was, how is this impacting, this other important thing at the time was the microbiome. And I think it was relatively new. And so that's really what, kind of sprung the whole interaction.
All right. So the contaminant at the time you already mentioned was the PFOA. I don't know if everyone's familiar with that as PFOA, but under that PFAS kind of terminology, could you tell us a little bit about it and maybe, where it's used and what it's used for, maybe where it shows up?
Over the last several years, we've been reading a lot about it in just the, general, newspapers, magazines, because it's been showing up in humans at fairly reasonable levels. I don't think we have a good understanding of at what level of the toxicity occurs. There's certainly some concerns about it because, they would not expect them to be, as pervasive throughout humans, but also I would say probably fairly easy to say that any organism on Earth probably has some level of these chemicals residing in them. So where do they come from? Anything that's going to be used for water repellents, plastics, anything like our cell phones, all of these things that we utilize in our day-to-day lives require these chemicals, for their production. Certainly, I think there's a big wish to move away from them in terms of, waterproofing and that sort of thing, non-stick, all that sort of stuff as well. But the problem is that they're really persistent in the environment. They don't break down as readily. And so I think that's why we've been seeing them in our bodies over the last, I think it's a question about when. It's been probably many decades, I think, that we know that they're there. And, there's been a lot of work to show that samples collected prior to their production, nothing is there. So this isn't some accident of nature. This is clearly an activity of humans. So they've been huge efforts now to comprehensively profile them. So this now, I think where people were just looking at a few, so PFAS, PFOA, now I don't even know what the number is now, but probably dozens, and they're metabolites too. So I think that's raised a lot of questions. And then, I think there's a lot of issues with, if we try to think about these as being foreign chemicals, we should probably keep them, to almost zero levels in us. And the fact that now pretty much everybody on Earth has them, and they're finding them in places that are so remote. It's just being shown that they're being carried, by a variety of different, atmospheric processes, that sort of thing. It's raised a lot of concern. And the question then is, what are the long-term health implications of this? And I think I don't think there's been a clear connection yet. A lot of it's been done through animal models, and then of course maybe through epidemiological studies. But I think this is, something that we'll begin to figure out here in the next, few years about what this means long term. And I think that, the question about people are certainly afraid of it. So I think we just got to figure out what it really means in terms of, are these exposures causing problems or not? I don't think we really know the answer to that yet or not.
How does PFOA impact human health? So it's still an unknown at this point. There are conjectures, right? There's theories and models that maybe are out there, but there's no clear evidence. Am I understanding that correctly on how it affects humans?
Yeah, I'd say it's not totally murky. I think we have a sense that there's some impact. What I don't think is clear is like the exact mechanism by which things may cause harm. And more importantly, I think with respect to toxicology, it's like an understanding of, what the dose is and what a typical dose is for you and I, and how that actually translates into, what we would consider, something that would cause toxicity. So, this has always been a big, central tenet of toxicology, and that's the dose makes the poison. And so it's something I think we need to wrestle with. But I think I would go as far as to say we're just not exactly 100% sure of what these long-term health implications are.
There is some epidemiological evidence that tends to increase disease outcomes, but again, those are only epidemiological data. with plenty of compounders. Either people are exposed to PFAS or they're exposed to other industrial poisons. So the question is, as Andrew mentioned, what is the causative mechanism? And we don't, it's my knowledge, we still don't know that. Those molecules are designed to be incredibly stable and non-reactive. So they're by design that don't interfere with life chemistry very much. But of course, when they start building up at high abundances, you don't know what would happen.
Yeah, and I think the big question is that it's hard to pinpoint one chemical is causing the problem. So as Costas said, it's much more complicated because we are exposed to it's a mixtures problem. And so what do all those mixtures of chemicals actually do? And is the mixture that I'm exposed to similar to what you guys may be exposed to? And it could be specific to the individual being, that the exposure is occurring to. And of course, all the other things, the big one is, of course, diet, health, exercise, all these other factors play an important role in this. And so we're, trying to disentangle that. It's easy to do in mice, right? Because we can control them really easily. We know what we feed them. We know how active they are. We can even control, some of those aspects really relatively easily. But for, your free-ranging human, that's really challenging to do.
How have you seen this effort grow and mature since it started? Again, we're looking back like a decade. Some of the things that have changed and or grown.
Yeah, I think one of the interesting observations we made early on with this and a variety of other environmental contaminants was their impact on bile acid metabolism. So these are molecules that, if we look in our textbooks, are important for fat solubilization after you eat a meal. But what we quickly realized was that, these were a good readout of changes in microbial activity, enzymatic activity in particular. And that was one of the questions I actually had brought up to Costas's group was, we can see these changes occurring in the bile acid pool. In particular, we could see changes in the way bile acids are decorated with different amino acids. And so one of the things we began to wonder was why? What is going on here? And for probably decades, people just thought this was a way to reduce their toxicity. So microbes deconjugate, they're removing amino acids from them, in this case, glycine or taurine. And the big question was, well, why? And I couldn't really get a clear answer from that by looking in the literature. So then I had talked to Costas about this, and Costas, I believe it's Joshua. Is that right?
Joseph Chan, yes, my former postdoc, is now an assistant professor at Colorado State.
Yeah. So we started working together to try to figure out what this meant. And actually, through this collaboration, Joshua was able to find some really interesting hypotheses that were outside of kind of the toxicity question. So really having to do with perhaps that, the microbes could utilize these as energy sources. And I think that was where it very quickly, morphed into this, understanding of like how our microbes are then responding to these changes, and then more importantly, how can we model this? And that was really hard. I mean, it's not something my group is at all familiar with, but the expertise that we had, with Costas's group really allowed us to address this question. And then Costas, I think it was after this paper, then we worked with Gary Wu more on that neonatal thing. And this is again the same type of thing. Like, how do we begin to integrate the changes that we see in the microbial community from, say, just like if we're just counting who's there, versus what we see when we measure their metabolites through metabolomics? We didn't have a really good way of integrating that data. And that's where, our interaction and collaborations with Costas' group had a huge impact, because that started to allow us to put these pieces of the puzzle together in a way that actually, made some sense. So that was a really, tremendous, finding that we found through, this collaboration. I think that's, kind of where things have gone from this initial seed grant.
Yeah, Joshua, he did a very nice work. He developed metabolic models for many of the participants in the microbiome, the microbes that are present there. And he asked the question, which carbon molecule can be consumed by what microbe? And then he ended up generating a hypothesis as to, who is consuming what carbon molecule and why, what purpose. And he continued on the same path of modeling microbial communities. And right now he's running his own lab. He's assistant professor at Colorado State. So I think that had an impact in his career.
Where has this Sea Grant led you? And can you share some success stories?
This work that Joshua helped us with, obviously led, to in part to his landing a faculty position. We also published a number of papers. One was in M Systems. We subsequently, Costas and I then focused on more around in this case, not so much the environmental contaminant aspect, but focusing on more of the mechanism of how the microbes interact with host metabolites. So we wrote a grant with some partners at NIH to look at, in this case, this is a more basic question around how this whole interaction occurs. So that was a win for us. And I think now, coming full circle, we started working back on some of this perfluorinated work. And because of some of the insights we had originally, now we have some really amazing observations. It raises some important questions, and that is this exposure to these perfluorinated chemicals shifting our microbiome in a way that may be good or bad? We don't know. I'm not saying that it's a health benefit of this exposure, but we don't really necessarily know what the change in that composition of that community. It may make you susceptible to certain things like inflammatory bowel disease, right? This is hugely problematic here across the westernized world. And so exposure to things like this could trigger this. And I think this is something that, from a toxicology standpoint, we've not really fully considered over the last, I'd say, many decades, thinking as microbes as targets or toxicity endpoints. There's no, like, I can look in my, toxicology textbooks and there's no section on the microbiome. I mean, I think this is something that probably will be added because now we appreciate that this is really important, and we need to have a better understanding for how this whole interaction occurs. And I think this is which is super exciting about the field right now.
What are some next steps for the research, and maybe looking at things that you're either excited about or even maybe some challenges?
Yeah, I mean, I think as I alluded to before, I think one of the things that we're really interested in is thinking about how these persistent chemicals are perhaps transiently or maybe even permanently altering the microbiome depending on the exposure window. So one of the big questions people have is, if you're exposed early on in life, at birth, does this perhaps set you up in the wrong trajectory, if you will, health-wise? And there's been a lot of evidence with that with antibiotics. So antibiotic exposure, right? So that's, they know there's good evidence to show that this causes problems. The same can be true for these chemicals, right? So, they could be, bactericidal, bacteriostatic, and they can have the same properties, or, we may not be taking them for therapeutic purposes, right? But if they're manipulating or altering the community in a way that sets you up in a, kind of in a problematic way. I don't want to say dysfunctional because I don't really, it's hard to say what that actually means, but it is perhaps a contributor to some of our more modern health problems. I mean, I think that's, something that people are trying to tease out. So I'm excited about that because I think here, Penn State, we have the tools to do this. We now are doing really cool imaging studies to look at the individual bacteria themselves. This is something I never really would have thought to do, but was only I only learned about it through talking to someone at a networking thing one time where we were just sitting around talking about projects, and they said, "Oh, you should try this." So that's something that we've begun to explore now. And that's actually trying to image bacteria and to see what happens to them after certain exposures. And so you can do this by mass spec imaging now. There's other platforms to do that. So I'm super excited about that. And I think, because we have all the right connections, and we have a track record of doing good stuff with everyone here. So I think that's really sets everyone up for success. And I gotta say, I think the big thing is the excitement really builds around seeing the students get excited about their projects. I mean, I could speak for myself. Maybe Costas will agree. It's like most of the time I spend in my office, not doing very much in the lab at all. But to hear them come in and share something exciting, I mean, I think that's probably the best, most rewarding aspect of our jobs, is to see them get excited about stuff.
Yep, ultimately the students drive the projects. We're there to offer the occasional good idea, but they are the ultimate drivers of success or failure in all those collaborations.
How did early support from IEE enable success in this project?
One of the things that they were really adamant about was that you worked with someone outside of your college. And so that was, I think, another reason why Costas and I ended up working too, not just because of that reason, but I think that's made it a good fit. And, I think that really gave us kind of well, provide us a really good complementary collaboration where, we brought really, both groups brought a lot of strengths to the table. And because of that kind of caveat that was within the IEE solicitation, I think that was great. I mean, perhaps maybe there was some luck involved that we just happened to work together at the right time. But I think because of that push, that was really helpful. And I think that's something that IE is still championing. And I think other institutes, the Huck, for example, I know that they really favor that a lot, making sure that you're not just sticking to your home department, but that you're working outside of that. And then personally, it's just something that I really embrace, through our work as well, because I know that we're not experts in, everything, and that we do need help for doing things. And I think that makes things really fun. That makes research, really enjoyable.
Was there something that was surprising to you during this process that you learned or discovered?
I think one thing that's come from just this general question is that how it's probably a no-brainer to Costas, but to me it has been, and that was that how incredibly what good chemists microbes are. It's remarkable what we see, and in fact, a lot of the things that we measure in our mass specs, we can't annotate because they're not in some library, but we have a really strong feeling and maybe even a good appreciation that a lot of what we're seeing is not through host mediated mechanisms, but probably through microbial. So I would say that's been maybe the biggest surprising thing, to me.
Yeah, my microbes do bring to bear so many more enzymes than the host has. A mammalian host has. So yeah, if you think of really that matter, it's not surprising. You think of a pound or so of microbes could do so much, but not every single one comes in with its own armory of metabolic functions and enzymatic functions. And my feeling is that we only understand part of them. I think you start seeing compounds that when you do your MS, for that you have no pathway that can explain them. So clearly there is more chemistry they can do than we know at this point.
If you had 20 seconds, you're in an elevator with U.S. Senator, you got to pitch this project because you think it's the next best thing and you want them to champion it. What's your elevator pitch for this?
Is it going to be an 80-year-old senator? If that's the case, then I will tout the potential of this work towards extending lifespan, manipulating their microbiome in their gut and understanding how that can have a age prolongation effect.
Yeah, and I would echo the same thing. I think that, there's a lot of opportunities for drug development, trying to understand how they may be producing important drugs because they produce chemicals that manipulate us really, really well. There's lots of evidence of this. And so if we can tap into that and get a better understanding for how this occurs, there's, huge rewards there from a, therapeutic standpoint to promote our health, reduce disease, and I think ideally improve nutritional outcomes, trying to understand, what we're feeding us in addition to our microbes, like how does that impact overall health? So I think that's another key component there. And we didn't touch on today, but is nutrition and diet. Diet plays a huge role in shaping our microbial community. And so if we can get a better sense of how that's working, I think that this could have really profound implications for, human health. There's a lot of people studying some of these long-lived Japanese individuals who by and large live perfectly healthy lives well into their, early hundreds. And so why is that? And one of the tantalizing things is that this is a contribution of the microbes, preventing them from some really, problematic diseases. It's interesting. Certainly there's a lot to be learned. And I think, like I said earlier on, that's good for us because it just means that there's more things for us to continue to work upon.
Thank you for taking time and speaking with us. So Costas, Andrew, it was a great conversation.
If anyone's interested in following up, certainly my contact information is on the web, so you can always look me up.
All right, that wraps up our time. Thank you so much for being on Growing Impact.
All right, thanks.
This has been Season 5, Episode 6 of Growing Impact. Thanks again to Andrew Patterson and Costas Maranis for speaking with me about their project. To watch a video version of this episode and to learn more about the research team, visit iee.psu.edu/podcast. Once you're there, you'll find previous episodes, transcripts, related graphics, and so much more. Our creative director is Chris Komletic, with graphic design and video production by Brenna Buck. marketing and social media by Tori Indeviro, and web support by John Stavanger. Join us again next month as we continue our exploration of Penn State research and its growing impact. Thanks for listening.